Hi guys, so today we are going to be talking about chapter 12, which is the nervous system. We're going to go through a little introduction, and then we're going to go through the organization, both structural and functional. We're going to get into the details about nervous tissue, and then we're going to talk about action potentials and neurons. Hopefully you remember a little bit from muscles. And then we're going to go how through the signal transmits itself at the synapses, and finally talk about regeneration of nervous tissue. So we have two body systems that help control basically all of your body's activities. They are the ones that are in charge and help integrate everything going on to help maintain homeostasis. And of course, those are the nervous and the endocrine system. Remember, the nervous system is very fast to respond, but its effects are short term. The endocrine system is slower to respond, but the effects last a lot longer. And also remember that the nervous system can override the endocrine system whenever necessary. So neurology is actually the branch of medicine that studies the nervous system. Any imbalance in the nervous system, of course, is going to cause some sort of illness. So we have to do three main things in order to keep that happy, steady state called homeostasis. The first thing is a sensory function. We have to not only sense the changes inside our body, but we also have to sense changes outside of our body and our environment. Then we have to integrate all of this information, which our brain usually does, and the central nervous system, so that we can interpret the changes. Finally, a motor function is going to be to react to the changes, which will usually either be a muscle contraction or some type of secretion from our glands. It is organized, it's divided into two branches, the central nervous system and the peripheral nervous system. The central nervous system is the brain and the spinal cord. The peripheral nervous system is the cranial and spinal nerves, the ganglia, and the sensory receptors. So the central nervous system goes down along the axial skeleton, and the peripheral nervous system is on the appendicular skeleton, if you want to think about it that way. Functionally speaking, the central nervous system is integrative. So what it's going to do is it's going to take all of the information that it gets, all of that sensory information, and it's going to determine what needs to be done, and then of course generate a response. The peripheral nervous system is the in and out, so to speak. So it's the sensory information coming into the central nervous system, and then it's the motor function going out of the central nervous system. So the peripheral is going to be the transmitter, so to speak. So it's going to transmit the information to the central nervous system and then transmit the information from the central nervous system. So there's both a sensory component and a motor component here. The sensory component is afferent, it is incoming. So it's going to have a lot of different receptors that are going to detect changes in the environment or changes in our internal body. We can have general receptors or we can have specific sense receptors. We will talk about all of those in the second semester of anatomy. Then there's the motor component. This is the efferent or efferent, outgoing. Think of E for exit. It's outgoing, it's exiting. And this is what conducts the impulses from the central nervous system out to the muscles and glands. So it's basically the response. So whatever response the central nervous system comes up with, the peripheral nervous system, the motor component, is going to take that response and send it where it needs to go so that it can happen. So there has three subdivisions for the peripheral nervous system. Central nervous system is just that. It is the brain and spinal cord. But the peripheral nervous system is further subdivided. The first subdivision is called the somatic nervous system. This is voluntary and it is under conscious control. These are basically our muscles. So whenever we want to move, that's our somatic nervous system happening. And we pretty much control when we want to move, and it is completely voluntary on if we want to move. There are sensory neurons, again, that are going to conduct impulses from our special sensory receptors and the cutaneous receptors in our skin to the central nervous system. And then there are motor or efferent neurons that are going to send impulses from the central nervous system out to our skeletal muscles so that we can produce the movement. So basically, we know what we want to do, and sensory neurons are going to conduct those impulses to our central nervous system. And then motor impulses are going to be sent out, and they're going to go to our skeletal muscles, where an action potential will occur, which hopefully you remember from last information on muscles, and our muscles are going to move. The second is the autonomic nervous system. Now, the autonomic nervous system has two divisions. It is involuntary and it is unconsciously controlled. So we have nothing really to do with the autonomic nervous system. Just think of automatic. It is automatic. It automatically happens. So that has two subdivisions, which are the sympathetic and the parasympathetic. The sympathetic is our fight or flight, and the parasympathetic is rest and digest. And we're going to talk a lot more about those in chapter 15. Again, there are sensory or afferent neurons that conduct the impulses to the central nervous system, and then motor or efferent neurons that send the impulses from the central nervous system out to smooth muscle, cardiac muscle, and glands. So smooth muscle, like in our digestive system, cardiac muscle, which of course is our heart, and then glands that are in the endocrine system, which we will cover the endocrine system in the second semester as well. And then the third part is the enteric nervous system. Now, this is closely related to the autonomic nervous system and actually used to just be considered the autonomic nervous system. This is recently being taught as a separate nervous system. It is also involuntary, and basically it is our gastrointestinal tract, so our digestive system. The smooth muscle and glands that actually help us digest food through enteric plexuses. And again, we'll talk more about this later on. So this is a little animation of the nervous system and how it's organized. If you want to go through it, you have to just make sure that you're in slide mode view, and then you can click on it and of course be connected to the internet, and this will run for you. Now, moving on, nervous tissue. There are two types of cells in nervous tissue, neurons and neuroglial. Neurons are the main nerve cells. So neurons are the cells that conduct all of the functions of the nervous system. Neuroglial cells are cells that support the neurons, and we'll talk about those in a second. So the picture over here, you have a neuron, and you can see these little guys. These are the dendrites. Those get incoming messages. And then this is the cell body. Now the cell body makes the neurotransmitter or anything that the neuron particularly needs. So the dendrites are going to get the message. They're going to send it to the cell body, which is going to generate the action potential. Now the hillock is the trigger zone. That is where either the threshold is going to be met or it's not. If the threshold is met, then an action potential is going to be generated. If it is not met, then the signal will just go away. And then this is the axon. That is where the action potential is actually transmitted. So it's transmitted down the axon. Now the axon can be myelinated or unmyelinated. This picture is showing you a myelinated axon. So these little sheets here, those are sheets of myelin. And between every sheet is what's called a node of Ranvier. And we'll talk about that a little bit later. But basically, myelination speeds up transmission. And then at the end, you have the axon terminals. So that's where the signal is then going to be passed to another neuron or to the receptor, whatever it's going to, the target organ, whatever's going to make the change. And at the end are the bulbs. Those are where the neurotransmitters are housed, and they are released from these bulbs. So the cell body, the soma, has cytoplasm, nucleus, and various organelles, just like A lot of other cell bodies do. Nissl bodies are the rough ER. So rough endoplasmic reticulum, which hopefully you remember from biology, that's where proteins are made. So the rough ER has a special name in the neurons, and it's called Nissl bodies. And basically, it's going to produce the proteins for growth and repair. As I said, the hillock is where the action potential is generated, and it's basically where the cell body connects to the axon. Then the dendrites are the nerve fibers that extend out from the cell body. It receives input and brings it in towards the cell body. They're very short, they have a lot of branches, and they are not myelinated. And then, like I said, the axon, that's what conducts the impulses away from the cell body to either another neuron or the effector organ. It's very long, it's very thin, and it can be, like I said, myelinated or unmyelinated. The synaptic end bulbs, like I said, house the neurotransmitter, and that is where they are released from. So a nerve in general is a bundle of nerve fibers, which are your axons in the peripheral nervous system. So when you think of a nerve, think of a bunch of axons wrapped up together. Now between the nerve fibers is connective tissue. And we have 3 levels. We have endoneurium, perineurium, and epineurium. Now these should all sound kind of familiar from muscles and the coverings on muscles, except it was mysium instead of neurium. We have the endomysium, the perimysium, and the epimysium. Now it's the same exact thing, except with neurium instead, because we're talking about nerves. So here's a peripheral nerve here. Here's a myelinated axon. The Schwann cell is what myelinates the axon. It's what produces the lipids and actually myelinates it. The endoneurium is what covers the axons, okay? The perineurium covers a bundle of axons, which is called a fascicle. Now that should sound familiar from muscles. And then the epineurium covers the entire nerve. So the endoneurium is the smallest, covers the nerve fibers. Perineurium is a little bit bigger. It covers a bunch of nerve fibers, which is called a fascicle. And the epineurium is the biggest. It covers the whole nerve. Now we have cranial nerves and we have spinal nerves. Cranial nerves, there are 12 pairs, and we will actually cover that in the next semester. And then spinal nerves, there's 31 pairs, and we will cover that coming up soon. Now, you could have sensory nerves, you could have motor nerves, or you could have mixed nerves. Mixed nerves basically just contain both sensory and motor fibers. So a mixed nerve can get an incoming signal, take it to the central nervous system, and then bring the motor signal back out. In the central nervous system, there isn't any connective tissue between the nerve fiber bundles. So a bundle of nerve fibers or axons are going to be called a tract instead of a nerve. Okay, so a bundle of nerve fibers in the peripheral nervous system is a nerve, but a bundle of nerve fibers in the central nervous system is called a tract because it doesn't have that connective tissue surrounding it. Okay. Ganglia are just clusters of neuronal cell bodies in the peripheral nervous system. Nuclei are clusters of neuronal cell bodies in the central nervous system. So you're going to see a lot of differentiations between the names of things if it's in the central nervous system versus the peripheral nervous system. So we have nerves in the peripheral nervous system, but we have tracts in the central nervous system. We have ganglia in the peripheral nervous system, but we have nuclei in the central nervous system. So it's just a matter of remembering which is which. So, as I just pretty much said, a bundle of axon fibers in the peripheral nervous system is a nerve, but in the central it's a tract. A group of cell bodies in the peripheral nervous system is ganglia, but in the central nervous system it's nuclei. Now, neurons are what connects our body together. all regions of our body are connected to the brain and spinal cord through neurons. However, neurons have lost the ability to undergo mitotic divisions. So that can be a problem if you have damage to them, because neurons cannot regenerate themselves very much. We're going to talk about a special case at the end with the peripheral nervous system, but central nervous system, basically neurons can't regenerate. Now they can be classified by structure or function. Structural classification is based on the number of processes that extend from the cell body. So here's your cell body, here's your cell body, and here's your cell body. So you're looking at the number of processes. So a multipolar neuron has one axon coming from the cell body, but a bunch of dendrites. A bipolar has one axon and one dendrite. Now that dendrite can branch out, but the one coming off of the cell body is just, there's one dendrite coming off, okay? One axon coming off. And then finally we have a unipolar, which has an axon and a dendrite fused together coming from the cell body. So you've got this one process coming off, and then it's actually axons and dendrites fused together, okay? So unipolar, uni means one. You have one axon dendrite fused together coming off the cell body. Bipolar means two. You've got one axon, one dendrite coming off of the cell body. Now keep in mind that dendrite can then branch out, but as long as the main portion is only one, then it's bipolar. Then you have multipolar. Multi means many. So you've got one axon and a bunch of dendrites coming off the cell body. So what type of neuron is the motor neuron? This is the motor neuron right here. Is it unipolar, bipolar, or multipolar? Hopefully you said multipolar. You've got one axon coming off and a bunch of dendrites coming off. Now, what type of neuron is this interneuron here? This is the interneuron. Hopefully you said multipolar. It's got a bunch of dendrites coming off here and then one axon. So multipolar neurons are going to be located in the peripheral and central nervous system, and they are going to be motor neurons and interneurons, okay? Bipolar neurons, so you have the one dendrite coming off and the one axon coming off. These are specialized sensory neurons. So these are going to be in your special senses. So for seeing, hearing, smelling. And we're going to talk about those more when we get to the chapter on special senses, which is next semester. But this kind of gives you a preview of what they look like. This is the eye, and you can see we have different cell types here, and here's the pigmented layer of the retina, your rods and cones, which everybody's heard of, but then these specialized ganglion cells, bipolar cells, horizontal cells, and amacrine cells. Those are what we'll talk about when we get to that chapter. But remember, bipolar neurons are specialized for the special senses. So what type of neuron is this sensory neuron up here? Is this unipolar, bipolar, or multipolar? Unipolar. Why? Because it has one process coming off, and it's actually the axon and dendrites fused together. Now, unipolar neurons are going to be general sense receptors, so touch, pain, pressure, temperature. And we will also talk about those next semester. Functionally speaking, it is based on which direction the action potentials are going in relation to the central nervous system. So we have sensory, which are afferent. These conduct impulses from receptors into the central nervous system using the cranial nerves or the spinal nerves. So we're going from the receptors into the central nervous system, okay? So it's incoming, so afferent. Most of these are going to be unipolar. Motor or efferent neurons, efferent exit, so these are conducting impulses away from the central nervous system out to the effectors, which are usually going to be muscles or glands. Again, using the cranial or spinal nerves. These are always going to be multipolar. And then the interneuron is the integration neuron. So these are association neurons. These connect the sensory and the motor neurons inside the central nervous system. So those are going to be right here. That's the connection between sensory and motor. And most of those are going to be multipolar as well. So name the neuron, motor, sensory, or interneuron. We've got the sensory up top. Here is your receptor. Sensory is going to bring the impulses into the interneuron, which is the brain or spinal cord, which is then going to send a response out the motor neuron. Okay. This is going to introduce you to the nervous system. Again, if you want to play it, feel free. You just have to be in slideshow mode to be able to play it. So, neuroglia. These are also known as glial cells. These are smaller and more numerous than neurons. They are capable of division, but they are not electrically excitable. So basically what the glial cells do is support the neurons. Anything the neurons need, the glial cells are going to do. There are 5 different types found in the nervous system. Some of them are found only in the central nervous system. Some are found only in the peripheral nervous system. So in the central nervous system, astrocytes are star-shaped with a lot of processes, hence the name, astrocytes. So right here. They metabolize neurotransmitters. So when a neurotransmitter is released, astrocytes are going to break it down. They help maintain the potassium balance for the central nervous system neurons. And they help form the blood-brain barrier, which is very important to protect the brain. They also are a link between neurons and blood vessels. So in a nutshell, astrocytes are responsible for maintaining the environment for the neurons to make sure the neurons are happy. And that involves that potassium balance quite a bit. Then we have oligodendrocytes. Now, oligodendrocytes are right here. Now, they look like really tiny astrocytes, but they're very important because what they do is they produce the lipoprotein that forms the myelin sheath. Now, the myelin sheath is what is going to help speed up transmission on these axons. They form a network around the axons. So one oligodendrocyte can myelinate multiple axons, okay? And the reason I'm stressing that is because there's a difference between the myelin-producing central nervous system cells and peripheral nervous system cells, okay? And then we have microglia up here. Microglia are phagocytic. They basically are the cleanup crew and also help protect the central nervous system. So they act in immunity. Sorry. Very sorry. Okay, then we also have ependymal cells. Ependymal cells are simple cuboidal or columnar epithelial cells. They can be ciliated, but what they do is they line the brain ventricles and the central canal of the spinal cord. Very importantly, they produce cerebral spinal fluid. So that's their really important function. So those guys are right here, and they're going to be all over here. They're the lining, basically. But producing cerebral spinal fluid is very crucial to functioning in the central nervous system. Then we have 2 types of cells that are only found in the peripheral nervous system: satellite cells and Schwann cells. Schwann cells are right here. Now those are like the oligodendrocytes of the peripheral nervous system. Schwann cells are also called neurolimocytes, and they are basically flat cells that wrap around the axons in the peripheral nervous system. So they produce the myelin sheath around one axon. So they basically wrap around the axon kind of like a jelly roll. It'll just keep wrapping around it, and that forms the myelin sheath. Now, Schwann cells are only on one axon. They can't do multiple like the oligodendrocytes do. Schwann cells also have limited regeneration capability, which we're going to talk about later, but oligodendrocytes do not. Finally, we have satellite cells. These are flat cells that wrap around the cell bodies of neurons. We're pretty sure that they regulate the exchange of materials in and out of the cell body, but until recently, their function was kind of unknown. We knew they were there, but weren't really sure why. So we think it's a regulatory function. So here's an overview showing you the neuroglia, which ones are in the peripheral nervous system versus the central nervous system, and what they do. So like I said, we have two types that actually produce the myelin sheath, oligodendrocytes and Schwann cells. Oligodendrocytes are the ones in the central nervous system. Remember, they can form the myelin sheath around more than one axon. They do not regenerate. And we have myelinated areas, which are white matter, and we have unmyelinated areas, which are gray matter. So white matter, we're going to talk about white matter versus gray matter when we talk about the brain. So white matter is myelinated neurons, gray matter is unmyelinated neurons. Now, in between, there's those nodes of Ranvier I was talking about before. The nodes of Ranvier are the gaps in the myelin sheath. What that does is it allows transmission to be sped up because the action potential can jump from node to node to node. Now, in an unmyelinated neuron, it literally has to cover the entire neuron. But in a myelinated neuron, it can just jump from node to node to node. Okay? Now, Schwann cells, sorry, my dog is in here. Schwann cells are in the peripheral nervous system. They form the myelin sheath. Now, Schwann cells wrap around the axon like jelly rolls. They help regenerate the axon as well. And we're going to talk about that a little later. You have myelinated and you have unmyelinated. So myelinated neurons, again, transmission is going to be sped up because it can jump from node to node to node. Unmyelinated neurons, the action potential literally has to travel down the entire node. Okay? So it's like if you have 20 people standing in a line and you have to pass the marker to them. It's going to take a lot longer to pass the marker to each person going down that 20 line row than it would be if you had, say, 5 people and you could just throw it to the other person. Okay? So 5 people and throwing it from person to person to person is like a myelinated axon. 20 people and literally passing the marker from person to person to person is like an unmyelinated neuron. And we'll talk more about that later. So, myelination increases from birth to maturity. More myelination means better coordination. Myelin is just a lipoprotein, which is a lipid and a protein. But what it does is it electrically insulates the axon so that the electrical impulse stays on that axon and doesn't go other places, and also increases that conduction rate because, again, you can jump from node to node to node. Multiple sclerosis, or MS, is an autoimmune disease that basically what happens is the myelin sheaths in the central nervous system degenerate over time. Our body basically destroys the myelin sheath in the central nervous system. So it causes nerve impulses to kind of short circuit. So a nerve impulse will be traveling along and then a piece of myelin will basically be missing and it kind of short circuits it and cuts it out. It decreases the conduction rate. You have high CSF protein and there are cycles of attack and remission. So basically people with MS have good periods and bad periods. They'll have bad periods where they're constantly having these short circuits happening and they're having these attacks, and then they can be in remission for a little while. Guillain-Barré syndrome is an immune system response to a bacterial infection. Basically what this does is it causes macrophages to demyelinate the axons in the peripheral nervous system. Unfortunately, that results in acute paralysis. Most patients, however, have complete recovery because the bacterial infection eventually is cleared up. But while they have that bacterial infection, the macrophages are demyelinating those axons and paralyzing you in those particular areas. But again, once the bacterial infection is cleared up, the peripheral nervous system can remyelinate, because remember I said there's regenerative capabilities there, and patients can completely recover. So, white matter versus gray matter. White matter, again, these are the myelinated axons. So you're going to have bundles of nerve fibers called a tract in the central nervous system. The spinal cord, they're located around the periphery, and they have what are called ascending and descending tracts. Now, ascending go up, descending go down, as you can imagine. Gray matter are the cell bodies, so the nuclei, dendrites, axon terminals, and any unmyelinated axons in the neuroglia. The spinal cord, it's located in the center, shaped like a butterfly or an H. So if you look at the brain versus the spinal cord here, here's the spinal cord. The white matter is on the outside, the gray matter is on the inside. But in the brain, it's opposite. The gray matter is on the outside and the white matter is on the inside. Now just stop and think about this for a minute. Why would that be helpful? Well, because your brain, all of the stuff that's making the decisions and all of the tracks that connect it to the spinal cord are in the middle. So the white matter, which are where the myelinated axons are in the middle. On the other hand, the spinal cord, the purpose of a spinal cord is to connect the peripheral nervous system to the central nervous system, right? So the white matter, which is all of the myelinated neurons, are on the outside, which are going to connect to those peripheral nerves, okay? So the white matter on the outside of the spinal cord connects to all those peripheral nerves that we have going on, so impulses will be faster. And then it's going to send it to the brain, which in the center of our brain, that's where it's connected to the spinal cord. So it makes sense that all of the white matter is there because we want faster transmission. Now, when we talk about the brain, we're going to talk about how the brain was formed. And it wasn't necessarily formed with that in mind. But I think it's an easy way to remember where the gray matter and the white matter are, okay? So the brain forms an inner mass of brain tissue with white matter, a thin layer over the brain surfaces with the gray matter. Okay, action potentials, neurophysiology. Like I said, hopefully you remember something about this from muscle action potentials, because nervous action potentials are basically the same thing. When you have a muscle action potential, it is a nervous action potential that triggers it, okay? You have a neuron that is right at the neuromuscular junction that sends acetylcholine to that muscle to create the action potential in the muscle. An action potential had to get it there, okay? So first we need to talk about ion channels. Ions are going to move across the plasma membrane with their concentration gradient. So from high concentration to low concentration. Positive cations move towards negative and vice versa, opposites attract. When the ions move, they generate an electrical current that is going to change the membrane potential. And ions also use channel proteins to move across the plasma membrane. So here are pictures of the different types of channels that we have. But the point is that when those ions move, they change the membrane potential. So leaky channels randomly open and close. Sometimes ions leak out. That's why they're called leaky channels. Chemically gated are going to open in response to neurotransmitters, hormones, and ions. Mechanically gated are going to open in response to something mechanical, so a physical stimuli, vibration, stretch, touch, pressure, that kind of thing. And then voltage gated are going to open in response to that change in membrane potential, which is generated from the chemical or the mechanically gated ion channels. So again, changing that membrane potential is crucial to an action potential. So here's some leaky channels. You got the sodium and potassium leaks. So here, sodium can leak in, potassium can leak out. More membrane leaky channels are going to make the membrane more permeable to sodium and potassium. More potassium leaky channels there's more potassium leaky channels than there are sodium leaky channels. And potassium channels happen to be leakier than sodium channels. So potassium is going to leak out more than sodium will leak in. And that's going to cause an imbalance right there. Now, these are found in almost all areas and types of neurons. So as I said, potassium leaks out, sodium leaks in. Well, that should say in. I don't know why it says out. Sodium leaks in, sorry. Gated ion channels open based on the stimulus. So chemically gated is going to open based on a chemical stimulus, so a neurotransmitter, hormone, or ion. Mechanically gated are going to open in response to something mechanical, so physical, vibration, touch, pressure, stretch. Voltage gated in response to a change in that membrane potential. So chemically and mechanically gated channels will generate what are called graded potentials. Graded potentials are not full-blown action potentials, but they can add up to generate an action potential. Voltage-gated channels generate action potentials. So that's what's responsible for providing that excitability to muscles and nerves. So leaky channels randomly open and close. They're found everywhere. Chemically gated channels open in response to chemicals. They're found in dendrites of some sensory and like pain neurons, interneurons, and motor neurons. Mechanically gated, again, open in response to pressure, touch, stretch, something mechanical, physical. These are in the auditory receptors and your touch and pressure receptors. Both of those can generate graded potentials. Voltage-gated channels open in response to that change in membrane potential. They're found in the axons of all nerves, and that's what gives them their excitability because they generate action potentials. So we were just talking about changing the membrane potential. So what does that mean? Resting membrane potential is the membrane potential when the neuron is not conducting, okay? So when the neuron is not doing anything, it is at resting membrane potential. Positively charged are on the outside of the neuron, negatively charged are on the inside of the neuron. Resting membrane potential is negative 70 millivolts, and the membrane is considered polarized. So here's your membrane. Outside the cell, you normally have positively charged ions, so it's a positive charge overall. Inside the cell, you have negatively charged ions, so it's normally a negative overall charge. So you've got intracellular fluid, negative, extracellular fluid, positive. So resting membrane potential, again, is about negative 70 millivolts, and that's going to become important. So the membrane is more permeable to potassium than it is to sodium. So potassium leaks out relatively quickly, but sodium leaks in relatively slowly. The sodium-potassium pump right here is going to exchange 3 sodium out for 2 potassium in through active transport, meaning it requires ATP. But the sodium-potassium pump is going to become very important because that's what's going to restore resting membrane potential after the action potential has occurred. So sodium and potassium ion leak channels and the sodium-potassium pump are what actually maintain resting membrane potential. So as these potassium ions are leaking out and sodium is leaking in, that sodium-potassium pump is actively pumping sodium out and potassium in to keep the balance, in a nutshell. So what are graded potentials? Graded potentials are small changes from resting membrane potential caused by a stimulus. Hyperpolarized the inner membrane is more negatively charged than normally. Depolarized, the inner membrane is less negatively charged than normally. So hyperpolarized, you can have this little tiny negative dip. Depolarized, you have this positive change, okay? So less negative. This happens when those mechanically or chemically gated channels are opening and closing. So like I said, the mechanical and the chemically gated channels can cause graded potentials. If it causes the inside to be more negatively charged, it's called a hyperpolarization. If it causes the inside to be less negatively charged or more positive, it's a depolarization. So when the ions move through these gated channels, that's what's going to either depolarize or hyperpolarize the membrane. Okay. Now it also is going to depend on the ion charge and the flow direction, of course, because if you have positive going out and negative coming in, then nothing's really going to happen. But if you have more positive going out than you have negative coming in, you're going to have this depolarization. If you have less positive going out and more negative coming in, you're going to have this hyperpolarization. Okay, so graded potentials vary in size depending on the strength of the stimulus. The greater the stimulus, more channels are going to open, the bigger the change in membrane potential. So here we have resting membrane potential. If stimulus 1 is not very strong, you're going to have this small depolarization. Then if you have a stronger stimulus, you'll have a larger depolarization. And if you have an even larger stimulus, you'll have an even larger depolarization. So you'll have small graded potential versus large graded potentials. So the size of that graded potential, so the amount of depolarization, which equals the amount of excitation, is going to be dependent upon the strength of the stimulus. So the larger the stimulus, the larger the depolarization, the greater the excitation. Summation occurs when 2 or more graded potentials add together to cause an even greater change in membrane potential. So if you have 2 stimuli happening close together, what could happen is the membrane potential could be changing and depolarization could be occurring from stimulus 1, but not finished when stimulus 2 hits. So what that means is that'll kind of add and make an even greater depolarization occur. So the stimuli can be the same strength, but have a greater effect when they're closer together. Okay. So the graded potential is generated on membrane of the dendrites and the cell body, usually not axons. It stays localized, meaning it doesn't travel far. So graded potentials are going to be used for short distance communication only. It doesn't travel down the axon because it's not big enough. Okay, the stimulus isn't big enough, the depolarization isn't large enough, so the change in membrane potential is not big enough for it to travel. Okay. Now an action potential, however, is a sequence of rapidly occurring events that are going to depolarize the membrane, which will be followed by a repolarization. So an action potential is actually generated on the axons where these voltage gated channels are located. So these are where graded potentials are. These are where action potentials occur. These are good for either short or long distance communication. They follow the all or none principle though. Excuse me. Basically what that means is that an action potential has to get above a certain threshold or it won't happen, okay? So let's say there's a stimulus right here. Well, it depolarizes the membrane, but not much, okay? So the threshold is not met, depolarization is not going to occur, and an action potential will not be generated. However, if you have a larger stimulus that does depolarize the membrane enough, an action potential is going to fire. okay? So negative 70 is resting membrane potential. The threshold is negative 55. So the depolarization has to reach negative 55 millivolts or an action potential will not occur. So once it hits threshold, you can see the action potential fires. But like I said, it's all or none. So either it reaches threshold and fires, or it doesn't. That's it. Those are the only two options. Threshold met, depolarization occurs, action potential generated, or threshold not met, depolarization does not occur, no action potential. Now this is showing you what can happen when you have an extremely large stimulus here. So here's the two we just talked about. If you have a super large stimulus or a super threshold stimulus, the frequency of action potentials is going to be increased. So the greater the strength of the stimulus, the greater the frequency of the action potentials. So you'll have more action potentials firing with a larger stimulus. The stimulus intensity affects the frequency of action potentials, not the size of action potentials. It's very important to remember these numbers here. So negative 70 is resting membrane potential. Negative 55 is threshold. So that's where it has to meet for an action potential to fire. But positive 30 up here, that's as high as the action potentials go. The membrane will not get any higher than positive 30, okay? So the stimulus intensity is not going to make it go to positive 50. That won't happen. It will just create another action potential. So intensity affects frequency, not size, okay? Summation does not occur if the action potentials are generated, if multiple action potentials are generated, okay? So here we have our resting membrane potential. The voltage-gated sodium channels and potassium channels are closed, okay? Our membrane is considered polarized. Here's the stimulus. That causes the membrane to depolarize. If it reaches threshold at negative 55, we're going to have an action potential happen. So the depolarization phase, the voltage-gated sodium channels are open. Sodium ions rush into the cytosol inside the membrane is going to become less negative because sodium, which is positive, is rushing inside, okay? That's going to cause further depolarization, which is going to open even more sodium gates. We know this is positive feedback. The influx of sodium ions causes that depolarization to about positive 30 millivolts, okay? So everything's closed, sodium, potassium are closed. Stimulus hits, sodium channels open, sodium rushes into the cell, more sodium gates, more sodium into the cell, more positiveness, depolarization's occurring. Once it hits that positive 30, the voltage-gated potassium channels open and those sodium gates become inactive, okay? So now we're not going to have sodium coming into the cell anymore. The sodium channels are closed, so no more sodium coming in. However, those potassium channels opened, so we're going to have potassium channels going, or potassium ions going out of the cell, okay? So potassium's going out, nothing's coming in, which is going to cause the inside to become more negative because a positive charge is leaving, okay? So when the potassium channels open and the sodium channels close, the membrane begins to do what we call repolarize. So this is the repolarizing phase. So it's becoming more negative, going back down towards resting membrane potential, sorry. But what happens here is those voltage-gated potassium channels remain open a little too long, okay? So more sodium leaves, The inside becomes even more negative than resting membrane potential, usually about negative 90 millivolts. And then the voltage gated channels close. So potassium channels finally close, and then the sodium potassium pump kicks in and starts pumping 3 sodium out, 2 potassium in, until we're back to resting membrane potential. Okay, so just to recap, we have resting membrane potential, nothing's happening. A stimulus occurs, it hits threshold, sodium gates open, sodium rushes into the cell, causing it to become more positive or less negative. This is depolarizing. When it hits its threat maximum, so positive 30 millivolts, those sodium gates close, potassium channels open, so potassium starts leaving the cell, causing the cell to become more negative or repolarizing. However, the potassium gates stay open a little too long, too much potassium leaves, and the cell becomes hyperpolarized. It becomes even more negative, about negative 90. So the sodium potassium pump kicks in, pumps 3 sodium out, 2 potassium in, until resting membrane potential is restored. Okay? So action potentials always depolarize before you repolarize. Now, there's something called an absolute refractory period. This is the time when another impulse cannot be generated. This is basically during the depolarization and repolarization phase. So, during this time right here, it is the absolute refractory period. And what that means is that nerve cannot respond to another stimulus during that time. Now, in general, the frequency range between 10 and 1,000 per second. So keep in mind, it's really fast. The absolute refractory period does not last long because you have to remember how fast our nervous system is working. Okay? And here's some clips with overviews and action potentials for you to watch. So, graded potentials versus action potentials. Graded potentials originate in the dendrite or the cell body, and they stay there. Action potentials are on the axon. Graded potentials are chemically and mechanically chemically and mechanical gates, Action potentials are voltage gates. Graded potentials are short distances. They fade over time. Think about if you drop a pebble in water. Initially, the waves are kind of big, but the further away you get from that pebble, the smaller the waves. Kind of the same thing. Action potentials can be short or long. Stays the same over the distance. It doesn't dissipate over time. Okay? The amplitude of graded potentials varies. There can be big ones or smaller ones. Action potentials, it's all or none. Either they fire or they don't. Graded potentials are either hyperpolarization or depolarization. So they either get less or more negative or less negative. Action potentials always depolarize and then repolarize. Graded potentials can have summation occur, which if you have stimuli closer together, it's a higher potential. Action potentials actually have a refractory period where they cannot respond to another stimulus. So how do they propagate? Action potentials arise at that trigger zone, and then they travel down the axon to the axon terminal. We can have continuous conduction or saltatory conduction. Continuous conduction are on unmyelinated axons. Saltatory are on myelinated. Now, this is what I was kind of explaining before. Continuous conduction, the impulse literally has to travel all the way down this axon. So it's like if you have 20 people standing in a row and every person passes a marker to the next person. Versus saltatory conduction, where the action potential can jump from node to node to node. So that's like if you have 5 people in that same line and you throw the marker to each person. It's going to be a lot faster to do it that way. So the myelin sheath is that electrical insulator. It blocks membrane depolarization. So it'll be here, this is blocked, so it'll go there, this is blocked, and so on. Because it insulates it, that membrane cannot depolarize, forcing the action potential, the impulse, to jump, okay? The nodes of Ranvier, however, do have voltage-gated channels, so they can depolarize. The membrane depolarizes and repolarizes very fast, and then stops at the myelin sheath, so it jumps over, okay? So think of leapfrog, it leaps from node to node to node to node, and travels very fast. It also uses less energy, it uses less ATP, because only small amounts of the membrane are actually getting depolarized. So you have this part of the membrane getting depolarized, then this part, then this part. It's going to take a lot less ATP than if you have to depolarize this entire membrane, okay? Sorry if you hear my dogs barking in the background. So propagation speed is related to a few things. The larger the diameter of the nerve fiber, the faster the impulse is going to travel. Myelinated fibers are faster than unmyelinated, and warm nerve fibers conduct impulses faster than cool fibers. Stimulus intensity is due to the frequency of action potentials generated at the trigger zone, not the size. So remember, higher stimulus intensity, higher frequency of action potentials. So, signal transmission, what happens when it finally gets down the axon? Well, at the end of the axons, remember, we have these synaptic end bulbs. The synaptic end bulbs end, and then there's an area between another neuron called a synapse. Now, the synapse is that functional junction between neurons or between a neuron and an effector, which will be a muscle or a gland. So we have different types of neurons based on transmission. We have presynaptic neurons and we have postsynaptic neurons. Presynaptic, think of pre means before. So presynaptic neurons are before the synapse. So they send the signal towards the synapse. Postsynaptic neurons, think of post as after. So postsynaptic neurons are after the synapse. So they receive the signal and then carry it away from the synapse. So here you have a presynaptic neuron. Here's the synapse. Here's the postsynaptic neuron. There's the target tissue. Okay, so your central nervous system sends out a presynaptic neuron. This is the neurotransmitter here, these little red balls. The neurotransmitter is going to diffuse across the synapse, and the postsynaptic neuron is going to receive it and then carry it to the target organ. We have three types of synapses based on how they transmit. Exodendritic is a synapse between an axon and a dendrite. So right here. Here are the dendrites, here are axons. Axosomatic is between axons and cell bodies. And axoaxonic is between two axons. Okay, so axodendritic, axon dendrite, axosomatic, axon soma or cell body, axoaxonic, axon axon. There are a lot more synapses than we actually have neurons, because remember, each neuron has multiple synaptic end bulbs, and each synaptic end bulb is going to have a synapse. So here's a clip for y'all to watch, but in general, we have two types of synapses. Electrical, which is found in smooth muscle, cardiac muscle, and between neurons in the central nervous system. There's two-way communication between cells. So what happens is an ionic current is going to spread through gap junctions from cell to cell. Now remember, gap junctions are the communicating junctions. They let ions pass through rapidly. Electrical synapses are faster than chemical. So we can quickly coordinate and synchronize the activity of a whole group of muscles or neurons. Okay, so like in a heart, we can very quickly coordinate activity here because the gap junctions allow the ions to travel from cell to cell to cell very fast. They flow in two directions, so they go either way. The other kind is the chemical synapse. This is slower, but this is like your neuromuscular junctions. One-way transfer of information, so it goes from presynaptic to postsynaptic or the effector. Okay, so here's the presynaptic. These are the little vesicles of neurotransmitter. So the vesicle will fuse to the end of the synaptic end bulb. Neurotransmitter will go out into the synapse. It will diffuse across the synapse and attach to the postsynaptic neuron, which will then carry the information wherever it has to go. So presynaptic neuron end bulbs release the transmitter. Postsynaptic neuron receptors attach to that neurotransmitter. And the synaptic cleft is the synapse between them. Okay? Now these can be named based on the neurotransmitter released. If they release acetylcholine, like at the neuromuscular junction, they are cholinergic. If they release norepinephrine or epinephrine, they're called adrenergic. Okay, so cholinergic release acetylcholine, adrenergic release epinephrine or norepinephrine. So presynaptic neuron is going to release a neurotransmitter, which diffuses across the synaptic cleft, binds to the receptors on the postsynaptic neuron. So an exodendritic synapse would be axons and dendrites, remember. So the action potential is going to travel down the axon of that presynaptic neuron. It's going to release a neurotransmitter that will diffuse across the synaptic cleft. The receptors on the postsynaptic neuron will bind to that neurotransmitter and then send the action potential where it needs to go. Okay? So this is a chemical synapse because it's a neurotransmitter, which is a chemical that is signaling the response. So a postsynaptic potential is the type of graded potential caused by that postsynaptic neuron binding to the neurotransmitters. Okay, so here's the original action potential. It releases a neurotransmitter, it diffuses across the synaptic cleft, it binds to the dendrites on that postsynaptic neuron, which is then going to create a postsynaptic potential. Okay? So here we have the presynaptic axon terminal. Remember, this is the neurotransmitter. Binds and fuses to the membrane, releases the neurotransmitter, diffuses across the synaptic cleft, binds to the postsynaptic neuron, which will then create a postsynaptic potential. So here's your action potential. It reaches the synaptic end bulb. Calcium diffuses in. With this calcium rushing in, that's what actually causes the vesicles to fuse to the membrane and the neurotransmitter to be released. So calcium is important here because when the membrane depolarizes, it causes that calcium to rush into the cell, which is then going to allow the neurotransmitter to bind to the membrane and release its contents through exocytosis. Okay, so action potential gets down there, calcium channels open, calcium goes in, vesicles bind to the membrane, release the neurotransmitter, binds to the postsynaptic membrane receptors, causes either a hyperpolarization, which is an inhibition, it will stop the next neuron from doing something, or depolarization, which is an excitation, which will cause the next neuron to do something. It depends on which ion channels are open. Now, if the postsynaptic potential reaches threshold, it will then trigger an action potential down that axon. So just to recap, action potential comes down the presynaptic neuron. That depolarization causes these calcium gates to open. Calcium goes in. That causes the neurotransmitter vesicles to bind to the membrane and release the contents through exocytosis. The neurotransmitter binds to the postsynaptic membrane, Then that binding will cause either a hyperpolarization, so it will stop whatever it's doing, a depolarization, so it will start and keep doing whatever it's doing, or if it's high enough, an action potential will then go down this postsynaptic neuron. Okay, so you basically have three results. The postsynaptic neuron can be inhibited, it can be excited, or an action potential can occur and it will carry it even further. Okay. So we have excitatory and inhibitory, as I just said. Excitatory is going to depolarize that postsynaptic neuron. The inner membrane is less negative. Cations will either come in or anions will leave. This is going to generate what's called an excitatory postsynaptic potential, or an EPSP. And remember that EPSP, okay? It's closer to threshold potential. A single EPSP, however, is not enough to create an action potential. It's just closer to threshold than an inhibitory is, okay? The next EPSP may be enough to start it through summation, though. Remember, graded potentials can go through summation. So what this is all saying is an excitatory postsynaptic potential is closer to threshold, which means that if an impulse is a graded potential happens, okay? So we have one stimulus going in causing this graded excited potential. If another stimulus comes in fast, since it's already close to threshold, an action potential can actually be generated through summation, okay? And inhibitory hyperpolarizes the postsynaptic neuron. So the inner membrane is more negative. So anions come in or cations leave. This generates an inhibitory postsynaptic potential, or an IPSP. This is further from threshold, so an action potential will not be generated here. Okay, so if an excitatory postsynaptic potential is generated and another stimulus comes in really quick, an action potential can be generated through summation. But even if it's not, a single EPSP is going to cause the neuron to do whatever it's supposed to do. On the other hand, inhibitory hyperpolarizes it and makes it more negative. So it's going to just stop the neuron from doing whatever it's supposed to do. So it's going to inhibit the neuron. An action potential will not be generated because the neuron's been inhibited. Okay? And sorry for saying okay all the time. Okay. So each presynaptic neuron can release several different neurotransmitters simultaneously. So it's not like a presynaptic neuron can only release acetylcholine. It can release multiple neurotransmitters at the same time. Each postsynaptic neuron is specific for its neurotransmitter, though. So acetylcholine will only bind to acetylcholine, for example. The same neurotransmitter can be excitatory in some locations and inhibitory in others. It really depends upon the type of receptor, okay? So if a receptor is binding to acetylcholine and it happens to be an inhibitory receptor, that's going to cause acetylcholine to make it stop. On the other hand, if it's an excitatory receptor, that will cause the acetylcholine to make it start, in a nutshell. Neurotransmitters have to be removed from the synaptic cleft quickly, though, because we can't just have neurotransmitters hanging out. Diffusion out of the synaptic cleft can happen, meaning it just moves away from the synaptic cleft. Enzymes can actually degrade it. Acetylcholinesterase breaks down acetylcholine, as you can imagine. The ASE tells us it's an enzyme. Acetylcholine tells us it breaks down acetylcholine. Or it can be taken back up into the cells. actively transported back into the neurons, reuptake, or into the neuroglia, which is just uptake. Now, SSRIs are based off of that reuptake. Selective serotonin reuptake inhibitors, or SSRIs, prevent reuptake back into the neurons. So it leaves serotonin out in the synaptic cleft for longer so that our neurons have access to it for longer. So if you have a deficit in serotonin, you can take SSRIs like Prozac, for example, and the serotonin will be out in the synaptic cleft longer so your neurons can actually have access to it. And that treats depression and things like that. So each postsynaptic neuron receives input from a bunch of presynaptic neurons. So like you can see here, here's your postsynaptic cell. You got a bunch of presynaptic cells binding wherever they can. Some presynaptic signals are going to be excitatory and cause EPSPs. Some are inhibitory and they're going to cause IPSPs. But the sum of all of the messages that come in are going to tell the postsynaptic neuron what to do overall. So basically, if there's more excitatory impulses coming in than inhibitory, the overall message is the postsynaptic neuron is supposed to be excited. On the other hand, if there's more inhibitory than there are excitatory, the overall message is that the postsynaptic neuron should be inhibited. Like I just said, if EPSPs are greater than IPSPs, the postsynaptic neuron will be excited and depolarized. If that depolarization reaches threshold, an action potential will be generated. If IPSPs are greater than EPSPs, the postsynaptic neuron will hyperpolarize, no nerve impulse will be generated, and the postsynaptic neuron will actually be inhibited. So we have a few neurotransmitters associated with the central and peripheral nervous systems. A neurotransmitter is basically just a molecule released as a result of a nerve impulse. It comes from the axon terminal. It's released into the synaptic cleft, and it diffuses out of the synaptic cleft, binding to that postsynaptic neuron, which results in a change of membrane potential in that postsynaptic neuron. So one big one is acetylcholine. Cholinergic neurons, remember, release acetylcholine. They're in the somatic and autonomic nervous systems, and in both the sympathetic and parasympathetic. Excitatory is at neuromuscular junctions. So at a muscle, it's going to excite that muscle. Inhibitory on parasympathetic fibers to the heart, so it would slow down the heart rate. So acetylcholine can basically excite your muscles or slow down your heart. Pretty versatile. Then we have amino acids. Excitatory in the central nervous system like glutamate and aspartate. Inhibitory in the central nervous system like GABA in your brain and glycine in your spinal cord. Then we have biogenic amines, which are both excitatory and or inhibitory depending on the type of receptors at the synapse. In general, they're responsible for our sleep and wake patterns, moods, emotions, how we feel. You have norepinephrine and norepinephrine, also called adrenaline and noradrenaline. These are also hormones, but they do a dual role as neurotransmitters as well. Also dopamine and serotonin. Anginergic neurons in the sympathetic division of the ANS and catecholamines, which are synthesized from amino acid tyrosine, are going to pick up norepinephrine, epinephrine, and dopamine. An imbalance in these particular neurotransmitters is linked to depression and bipolar disorder, especially serotonin. That's a big one, which is why we have an entire group of medications called selective serotonin reuptake inhibitors to treat depression. There are some others, purines like ATP, ADP, AMP, nitric oxide, CO, neuropeptides. They can be excitatory and inhibitory, but these are not as important as the rest, basically. Some disorders, Parkinson's and schizophrenia is linked to a dopamine disorder. Alzheimer's has actually been linked to acetylcholine on the plasma membranes. Depression, bipolar, seasonal affective disorder, serotonin, norepinephrine, and dopamine. As I said, selective serotonin reuptake inhibitors like Prozac, Zoloft, and Paxil are going to leave serotonin out in the synapse for the neurons to be able to use, and they're pretty effective in treating it. Treatments of faulty neurotransmitters, you can produce a drug that mimics that neurotransmitter and block transmission of the neurotransmitter. Things that modify the effects of neurotransmitters, synthesis. Synthesis can either be stimulated or inhibited. Release of the neurotransmitter can either be enhanced or blocked. The receptor sites, they can be activated and ready to bind to the neurotransmitter or be blocked so they can't bind to the neurotransmitter. And then removal from the cleft. They can be stimulated to get the neurotransmitter out of there or inhibited like SSRIs to keep the neurotransmitter out there for longer periods of time. So Parkinson's, we have a decreased synthesis of dopamine, and that is actually treated with something called L-DOPA. L-DOPA is basically a synthetic dopamine. So if you think about it, if you have a decreased synthesis of dopamine and you treat it with a synthetic dopamine, it should do the trick. People have different success with it though. Amphetamines enhance the release of dopamine and norepinephrine. And botulinum toxin actually blocks the release of acetylcholine at the neuromuscular junction, causing paralysis. Now, that's a problem with Botox injections. If you have Botox injections, you have to make sure that you go to someone who knows what they're doing. Because if you do not have an avirulent strain of the botulinum toxin, you can actually die. And people have died. There was this party of 6 women, I believe it was. They had a Botox party, but they just hired a guy who had no idea what he was doing. And he actually ended up injecting a virulent strain of the botulinum toxin into them. And they all became paralyzed and died because they couldn't breathe. So if you're going to have Botox injections, just make sure you go to somebody who knows what they're doing. Don't be scared to have them because there are a lot of people that know what they're doing, but just please be sure they know what they're doing. Curare is a poison. It blocks acetylcholine receptors at the neuromuscular junction, causing muscle paralysis. Women used to poison their husbands with curare back in the day. It's a horrible thing, though, because basically they suffocate because they can't breathe and they are paralyzed and can't do anything about it. And then cocaine inhibits dopamine reuptake. So dopamine is left out in the synaptic cleft for longer periods of time, and you get a sense of euphoria because of that excessive stimulation of the brain from the dopamine being left out in the synaptic cleft. Other things that modify membrane potential, increased excitability is going to reduce the threshold and make it easier to depolarize the membrane. Caffeine and nicotine can do this. It can cause alkalosis. The pH of your blood is 7.35 to 7.45. If you have a pH higher than 7.45, you have alkalosis. It can cause numbness, tingling, lightheadedness, and even spasms, and should be treated. Hypnotics, tranquilizers, and anesthetics decrease excitability. So basically, it's more difficult for the membrane to depolarize because the threshold is increased. So you need more things to be excited. This can cause acidosis, which is a pH of less than 7.35. Weakness, lethargy, and even coma can be caused by this. So again, this should be treated. Unless, of course, you're taking an anesthetic because you're having surgery, then don't worry about it. Neuronal circuits, we have neurons organized into circuits. Keep in mind, we have billions of neurons in our bodies, even more synapses than those. They have to be organized in some type of way. So we have different types of circuits here. You can see we have diverging circuits, converging circuits, reverberating circuits, and partial after discharge circuit, or parallel after discharge circuits. So diverging, you have one main one, which diverges into a lot of outputs. Here you have a lot of inputs going into one output. Here you have inputs kind of reverberating and going back to create the signal over and over on an output. And here you have an input going out into parallels to all converge on that one output. If you have abnormal circuitry, you have epilepsy. So then we have regeneration of nervous tissue. At about 6 months of age, the neuronal cell bodies lose their mitotic apparatus, which means they cannot divide, they cannot go through mitosis. In the peripheral nervous system, myelinated axons and dendrites can be repaired, though, as long as the cell body and the Schwann cell are not damaged. So this is why, for example, if you cut off your pinky, they can surgically reattach it because those neurons will regenerate there. However, in the central nervous system, injuries cannot be repaired. Learning frequently stimulates production of new neurons in the hippocampus of the brain, but you cannot repair injured neurons. Okay, so there's a difference, which is why if you get a spinal cord injury, for example, and in the central nervous system it is severed, then you're basically going to be paralyzed from the site of the injury down because the central nervous system cannot repair itself. And that's the difference between peripheral nervous system and central nervous system damage. Schwann cells have that capability to regenerate as long as that cell body is intact. Oligodendrocytes do not. Okay? So plasticity is the capability to change based on experience. So when we learn new things, new neurons can be generated in our hippocampus, but that's it. Okay? When we learn new things, we also make new connections. So that's a big thing, making new connections, expanding those dendrites to connect to other neurons and using your neurons. That is one reason why I said repetition is the easiest way to learn. If you just keep repeating the information over and over and over again, you're going to learn it. I always say, think about your favorite song. You learn the words because, well, at least you think you learn the words, but you learn the words because you heard it so many times, not because you went and Googled the words and memorized them. You memorize them because you heard it over and over and over again, and probably even some songs you don't like. You've heard so many times that you know the words because of repetition. When I first started teaching, I used to have the PowerPoints like all spread out for me and all my notes in the margins, so I made sure I didn't, forget anything. But then after I was teaching for like a year, I didn't need those notes as much. And by the time I was in my second year of teaching, I didn't need the notes at all because I had repeated the information so many times that now I know what I want to say. Now I know what's important for you guys to know. So hopefully that makes sense. So that is it. Hopefully this helped you guys. And we will talk about more chapters in the future. Bye.